Welcome everybody to a new edition of our emerging topics in biomolecular magnetic resonance webinar. So we have two exciting speakers today from solution state and solid state NMR. Both speakers agreed to recording, so we will upload the talks to the ISAMBS YouTube channel. Just to remind everybody, please type in your questions into the Q&A and not the chat, or raise your hand and then we can unmute you and you can directly put forward a question to the speaker. Also to remind everybody that there's the early career researcher edition of the ISAMBS webinar, so please join there too. Without further ado, then I hand over the stage to Geoffrey Bodenhausen, who agreed to introduce the first speaker of today, which is Lyndon Emsley. Geoffrey?
Yes, thank you very much. So it is a great pleasure to introduce Lyndon. He came to my lab in Lausanne in about 87, 88. That's only about 2 years after I started a new group there. So I was quite naive and had no idea how to do this. And I asked him to work on liquid state NMR, although we also tried to do solids in parallel. It was not a very successful enterprise. He started working on NOSI in the presence of resolved couplings, which I think is still a brilliant paper, but never read by anybody. But then he switched to selected pulses, invented Gaussian cascades, and also quaternion-based cascades, so-called Q3 sequences, which I think are still reasonably fashionable. He applied these things to all sorts of cute selected experiments. There's a whole family of papers that go under the name of cis-COSY, also called cis-COSY, which means selective inversion something, and was then concluded his PhD in early 91. And he insisted on having among his examiners Richard Ernst. Of course, I had been working with Richard until 3 years before that, but I had carefully avoided re-inviting him. But Lyndon said we must have Ernst, so we did have Ernst, and he came. This was in March 91, a few months before it was announced that he got a Nobel Prize for Chemistry. If we had delayed Lyndon's thesis by a few months, it's quite likely that Richard would not have come for the event. So anyway, the thesis was a great success. Even Richard thought it was a good student. And then Lyndon went off to California and on his Miller Fellowship, starting in the summer of 91. So that's as much I would say today. He has become unfaithful to liquids and focused now on solids. Some years ago, we both got grants from the French government to buy an 800 megahertz wide bore and a gyrotron. And we had to decide at that point whether we should have an 800 with a cold head that would recycle its helium, or whether we should have a traditional one. And he chose the recycling one, and I chose the traditional one because I also wanted to do liquid state NMR. And I remember Lyndon saying, what is that, liquid state NMR? So he has apparently drifted away from liquids altogether and is now firmly entrenched in solids. Lyndon, up to you.
Okay, well, thank you very much, Geoffrey. I think one of the great things about this series that I'd forgotten is that they spring some eminent personality on you to introduce you. And so it's a real pleasure to be introduced by Geoffrey today, and that's some very nice old memories. So just let me start my screen sharing. And effectively, today I'm going to talk to you about solid state NMR. And the title is Drugs in Cells. It's thanks to this committee, Marcus and his gang, for inviting me to the Emerging Topics in BioNMR seminar, which isn't really what we do. I think that's what Geoffrey was inferring. So we don't do that much, protein-focused biological NMR anymore. But we are interested in bio-related problems, and I'd like to talk to you today about something new that we haven't shown anywhere else, and that we've just started working on more recently. And so that the topic today is going to be, drugs in cells. I know that some of you like this format. This is especially for Martin Blackledge and Lucio Friedman, if they're out there. So before I get to the actual news, the scientific news, in this format, I thought it would be a good idea to start with the weather, right? So let me just activate that. And so if you're living in Eastern Europe at the moment, you know weather is an issue. We're having a terrible summer, and you can see this big front of rain coming down through Germany at the moment, causing floods. That's 5 p.m. on Thursday. It looks like it's going to be getting better tomorrow, so we'll have scattered showers in the Lausanne region. The Germans are going to be seeing some better weather too. And it's nice to see that as we go into the weekend, we can see the skies will be clearing up over most of Western Europe, temperatures going up again. And finally, as we go into Sunday, it looks like we're going to have great weather for hiking in most of the Western Alps. So you can look forward to that. Okay, so that was the weather. Here's the news. So the question I'd like to talk to you about is, and this is driven by our partners. We have a long-standing collaboration now with the material science groups in AstraZeneca. And they've been asking us recently if it's possible to use NMR to detect drugs in cells. And the objective here, of course, is that they want to establish concentration response relationships at the cellular and molecular level of drug action. And you probably all know that there's been work on in-cell NMR over the last decade or so. Key figures working in that area doing great work. Most of it's centered on studying proteins in crowded environments or in cells, and proteins and protein interactions. There has been some work at looking at drug molecules, but we obviously wanted to think more about limits of detection, how we can detect drugs at relevant concentrations in cells, and maybe if we can localize them within the cell using NMR methods. And the reason that we're interested in this is because I think I'm going to be able to show you that DNP-enhanced NMR in the solid state might be the way to approach this. And so I'm very interested, looking forward to any debate at the end of the talk on what you think about this way forward. So why are we thinking this is promising? Over the last few years, my group and others have been developing DNP-enhanced magic angle spinning NMR methods to look at particle size distributions in materials, so in polymers or in drug formulations. And there's a very simple underlying idea here, which is that if you use impregnation DNP, so impregnation DNP would be to take a substrate, here an organic particle, to impregnate it with a polarizing solution, and that would be a solvent radical mixture, typically with the radical at a few, 16 millimolar or whatever. If we impregnate the surface of this particle with the polarizing mixture, and then we turn the microwaves on, what we'll do is we'll generate hyperpolarization in this surface, in this layer outside the particle. Okay, so that's the solvent enhancement that we're used to seeing if you're doing NMR in frozen solution with MAS-DNP. But what will happen now is that spontaneous spin diffusion will relay hyperpolarization from the surface into the particle. And so over time, we're going to see the particle becoming polarized, and so we'll see the signal from the particle growing up. Okay, and this is a process that's governed by spin diffusion. Okay, and it's quite a simple idea. And today we've developed some very sophisticated analysis methods using, again, these are all classical models for heat transfer, for example, which can be applied to polarization transfer, and which allow us to characterize, potentially in very fine detail, the composition and localized spatial localization of different components in complex mixtures. And this is work that we've been doing in particular recently with Brad Schmelke at UCSB to understand the models here. For what I'll talk about today, you don't actually need to go into that level of detail, though I invite you to take a look at that work. For what we're talking about here, in fact, it's actually a very simple idea, and there's a simple result. What we expect to see, for example, is if the substrate is localized in the polarizing solution, it will share the enhancement of the polarizing solution. It will be high. If in this, for example, model of a core shell system, if you're in the shell, you'll have an enhancement which is lower than the solution, because we relay polarization from the solution into the shell. We'll lose some polarization. And if you're in the core, so that's even further away, then you'll have an even lower steady state enhancement. And so if you look at these build-up curves, for example, this is just build-up curves. This is actually the enhancement as a function of time, for example. We would expect that in a homogeneous solution, the enhancement is constant as a function of time, and that the enhancement actually changes as a function of time in these two substrates in a way that's characteristic. So these curves are characteristic, the shape of the curve is characteristic of the structure that we're looking at. And then the steady state enhancements basically tell us the order, how far in you are to the particle. Okay, so there's a very simple approach, which is if we just look at the hierarchy of enhancements in a system, this in principle tells you how far you are away from the source. Okay, so I think it might be a bit more complicated than that, but that's a very simple approach to this that works most of the time. There are many examples of this now. So we've got several groups from our group, other groups have been doing on this. There's a very nice example from Mark Baldus early on, where he was looking at a layered system on biosilica with proteins and carbohydrates on the surface. And the example that I'd like to illustrate this with today is one that we did a few years ago in 2017, actually, on lipid nanoparticles. And of course, since we did this work, lipid nanoparticles have become much more trendy because you probably know that they are the delivery. This is how we do drug delivery for things like RNAs. And the mRNA vaccines that most of us have taken in the last few weeks or months are actually delivered using lipid nanoparticles. And so these are mixtures of lipids that form a small particle and which will carry the drug inside the particle. Okay, and the question, there's a lot of debate about the nature of these structures because they're difficult to study. Neutron diffraction is one method that's been used extensively, which I think tells you how difficult they are to study, because that's always a last resort kind of technique. And so the structure of these could have been, for example, an onion-like structure. There was some question that there was some water pools being formed inside the structure, or some kind of heterogeneous structure, or rather, this is the more homogeneous structure, and this would be a more heterogeneous layered structure. Okay, so the question is, do we have these layered type structures, or do we have more homogeneous structures in these materials? And so if you translate that back to from the DNP point of view, from the basic NMR point of view, this means that we're being asked to distinguish between two types of model. One is the so-called homogeneous core, okay, in which case the whole nanoparticle has the same structure throughout. And then the other model is the layered structure, in which you'd have one type of lipid or one type of some of the components which are at the surface, and some of them which are only localized in the core, in which case we'll have a gradient of concentrations. Okay. And so, for example, if we do DNP NMR on these systems, what we expect to see here is that all the components in the homogeneous model would have the same enhancement. And if we go to the layered structure, then we would expect to see higher enhancement for the components of the surface than in the core. Okay, so it looks like there might be an easy way to probe which of these two models is more representative of the actual structures. And so this is an example of the kind of data that we measure here. And so here we're looking at a comparison of DNP enhancements for each component in the multi-component structures. So we can measure the different lipids, the DSPC, the DMP PEG, the alkanes, and the cholesterol, the cationic lipids. And we can measure the enhancements. So we're looking at proton DNP enhancements, but which are measured by CP to carbon. So we can basically tag the position in the, or the component in the mixture through the carbon chemical shift, but we're always going to be measuring proton DNP enhancements. Okay, and if we take a closer look at the result that we see here, what we're seeing is that in the color here, we have the experimental measurement. And then in black, we have the prediction or the best fit for a layered model. And in gray, we have the best fit to the data for a homogeneous model. So as you see, the prediction for the gray, the homogeneous model, is that all the enhancements should be the same. Okay, and the prediction for the layered model is that, for example, the DSPC and the DMPE, which would be in the outer layer, would have higher enhancements, and the alkane and the cholesterol, which would be in the interior, would have lower enhancements. And we can clearly see that that's the model that this data supports. So the model very clearly supports a layered structure for the lipid nanoparticles. And then, of course, it's very interesting with respect to the questions that I was asking at the beginning. So if we now add the drug to this carrier, so we add this, in this case, it's a small interfering RNA, so siRNA. If we add the siRNA, then we can see where is the siRNA in the material, okay? And again, what we see here are the predictions that we have for the different models down here. We're measuring the RNA now through the phosphorus chemical shift, but always the proton enhancement transferred to phosphorus in this case. And so again, in this case, we see that the RNA has the enhancement that we would predict if it's sitting inside the alkane cholesterol part of the material. Okay, so this again shows us that the cargo is co-located with the components in the core of the LNP, and it is not present in the shell, the layer, the top layer of the LNP. So this is just what I hope is a nice example of showing you how, if things are working in our favor, if we can identify proper tags, if we have the right resolution and we have good enough sensitivity, then we should be able to locate substrates within structures based on either the steady state enhancement, which is what we're doing here, or the build-up behavior of the hyperpolarization as a function of time. Both of those should be tags for location. Which brings us to the second kind of problem, or the problem that we've been most concerned with, which is actually what's the NMR limit of detection? Can we actually go down to concentrations that would be relevant to drugs in cells? Okay, and the idea is not to load the cell with a drug artificially, but just to follow the action of a drug inside a cell at physiological concentrations. And if you think about this problem, this is one of the core problem in DNP, of course, is that if you take the base level of concentration, that's what we see here on the left for an organic solid, then about 300 micromoles in a rotor gives you an NMR spectrum in about half an hour. And if you go through different materials where you're reducing progressively the amount of material in the rotor, and for example, in materials, this would be going to, for example, a mesoporous material with a high surface area, well, you already have a 20 to 30-fold reduction in concentration. If you go to nanoparticles, you have a 30 to 50-fold reduction in concentration, you get lower surface areas. If you look at microparticles of crystals, 1,000-fold. And for example, one of the targets that we were aiming at was if you could do NMR of, for example, DNA on the glass plate, so of a DNA type sensor array, you'd be looking at a flat surface, very, very low surface area. You have about 300 picomoles in the rotor and in 40 milligrams of solid, and that would correspond to about a 1,000,000-fold reduction in concentration compared to our standard bulk samples. And so the question is, can we get a 1,000,000-fold reduction, signal enhancement from the DNP methods? And again, the short answer today is yes, you can, in the words of Barack Obama. So you go, this doesn't all come from DNP. DNP typically, you know, that will give you a factor 150, maybe 200. So let's be more generous, a factor ranging between 50 and 300, okay? But certainly not 1,000,000. But the other factors will come from, you know, intelligent sample preparation, so what we call impregnation DNP in our case, so you don't dilute the sample. We go to low temperature, so 100 Kelvin gets you a factor 3. There's some paramagnetic quenching in some samples, probably not in the samples that we're concerned with today, and that'll be working against us. We're going to go to nuclei which are abundant. So, you know, rather than looking at natural abundance carbon-13, we could look at phosphorus. That'll give us another factor of 100. We're going to use detection techniques that optimize everything on the NMR side. So CPMG, for example, or proton detection, another factor 3. And then we can look at how the molecules themselves can multiply the signals, and then maybe do it for a bit longer than half an hour. So in this case, 5 hours. And for example, if we combine all of those factors, we think we can get roughly a factor 1,000,000, so 6 orders of magnitude improvement in signal to noise with respect to basic NMR experiments. Okay, and this, what we're talking about here, the signal enhancement is not just the DNP enhancement, it really is the amount of signal you get in a reference experiment without DNP compared to the amount of signal in the best experiment you could do, without DNP. Okay, and so here's an example of doing that. This was done by Brennan Walder when he was in the lab, and it's that example of DNA and RNA on flat glass plates. So this would be mimicking DNA sensor systems. We use a label here on the phosphorus NMR. We thiolate the bases so that we get a very distinctive phosphorus chemical shift here. We can very clearly see the phosphorus chemical shift in both of these two different samples, and they correspond to samples which contain about 100 picomoles of the PS DNA strand in this case. And so we're actually looking, that's the duplex. And so we're looking at 80 picomoles of the DNA strand, which is attached to the surface and in conjugation with its conjugate. Okay, so we really can go down, it looks like, into this picomolar range for detection using DNP in NMR. And how does that compare to, I have trouble saying it these days, but Jeffrey brought it up, so I will talk about solution NMR. So yeah, how does that compare to solution NMR on a good day? Well, obviously, if you take solution proton NMR, then according to us in a 400 megahertz system today, the limit of detection for proton NMR is about 500 nanomolar, and that corresponds to about 300 picomoles in the sample tube. Okay, so obviously proton NMR in solution is a fairly sensitive technique. Okay, but if you think about it, that's not useful for drugs in cells because we're never going to be able to see a low concentration drug compared to the background in the cell. So we can't use proton. It's a bit like the same idea as hyperpolarized carbon imaging in dissolution DNP. Proton NMR is not the way to be able to follow those tracers. So if we want to see the drug, we're going to have to look at carbon-13, for example. Here's one model system that we've been looking at, this WNTC59 molecule, in which we were able to get carbon-13 labeled. So let's use carbon-13 solution NMR as our reference, and then we go down to about 60 micromolar, or about 30 nanomoles in the sample system. Okay, and of course that's not the experiment you do. The best experiment would be to do inept, and with inept, the limit of detection is about 20 micromolar, or about 10 nanomoles in the sample tube. Again, you can probably argue a bit about whether or not you can do better, but that's roughly where the limits of detection are today for solution NMR in carbon-13. When we measure so that's going to be our reference, and when we measure the sensitivity in the DNP MAS NMR of a frozen solution at 400 megahertz using CP to do cross-polarization from protons to carbon with an epsilon of about 100 in the solution, 12 kilohertz MAS, so these are experimental data, then we get a limit of detection which is actually much lower, which is 500 nanomolar. That's 10 picomoles of sample in the rotor. The sample holders are smaller. So actually, what we're showing here is that the most sensitive, according to us, the most sensitive way to detect an NMR signal, bulk NMR signal today using a commercial spectrometer is actually in the frozen solution using MAS DNP NMR. So that's interesting. And this is a kind of graphical, again, comparison of that. So we have a quite fairly low limit of detection for protons in solution. This is in terms of mass for the sample. And in fact, it's carbon in the MAS DNP that gives us the lowest limit of detection in terms of mass today in the standard systems. Okay, and the difference is actually quite large. So it's quite significantly better. The other marker that we're very interested in using, for example, is nitrogen-15. That might be even better than carbon. In carbon, we still might have problems with background. And so we've been very interested in these nitro groups as markers. And again, so here's another system that we've been looking at. It's a Kia 98014 with a nitroxy marker where we've labeled the NO2 group with the 15N. And if we compare limits of detection for 15N in a similar way in solution NMR, and here we're looking at direct N15 because we're not going to be able to do CP to this nitro group. Okay, so if we look at direct N15, then the limit of detection would be about 50 micromolars, 30 nanomoles in the sample. And then again, if we look at the DNP MAS NMR experiment in the frozen solution at 400 megahertz, we get an epsilon of about 150 in this case, and the limit of detection is about 2.5 micromolar or 50 picomoles in the sample. So again, we're down into that tens of picomoles in terms of amount of sample, limits of detection, and the micromolar range in terms of concentration. Again, that's just a summary of this, again, showing that the MAS-DNP experiment is the most sensitive way to detect this nitro group by NMR in a bulk sample, as far as we can see. Okay, so we have a very sensitive method of detection. Limits of detection seem to be where they would need to be to see drugs in cells. And so then we ask ourselves about quantification. We've been doing some work on quantification. We use fairly standard approaches here, using an internal standard. So we're going to add an internal standard to the polarization solution. This could be calcium formate, it could be imidazole, and then we'll have some analyte, which could be the drug, or here we've got something like pyruvate, or who knows what. Okay, and so we're going to use the internal standard to reference the analyte, and quantification is then quite straightforward. So there's a simple, it's a simple linear relationship between the peak intensities and the amount in the sample. And if you know the amount of the standard, then you can determine the amount of analyte. And of course, the only thing you're thinking about carbon-13 NMR, I remember at school, they told me that's not quantitative. And so quantification is possible if the intensities are quantitative. And I just want to remind you that today we have some very efficient ways of making carbon-13 NMR in solids quantitative. And so we've just taken the method that was recently introduced by Klaus Schmidt-Rohr in 2014, and which is a very robust way to do quantitative carbon-13 NMR. And we've just revalidated that here. If you compare multiple contact CP methods, and that's the approach that was suggested by Klaus, we find that the quantification errors are much lower than when we try and use variable CP methods, which were historically proposed, or if you just try and use a single cross-polarization time. But the simple message here is that if you use these multiple contact methods, which are very easy to implement, it's a very straightforward approach, and it allows you to homogenize magnetization over several CP cycles with a period of spin diffusion in between, and then we get very reliable quantification, even under these DNP conditions. So we can accurately quantify the amount of sample that we have in the sample. And this is true even when we have some more complex mixtures. So here we're looking at a mixture where we have the standard, we have the analyte, and they're in a liposome environment. And so the normal spectrum is dominated, or is rather polluted by the signal from the liposomes themselves. And we can actually just go ahead and, again, using standard approaches, subtract the signal of the basic liposome from the complex mixture, quantify the resulting spectrum, and here we measure 140 micromolar, so it's quite a concentrated solution. That doesn't really matter. The important thing is that we measure it accurately to within about 5% of the known concentration in this case. So we seem to be able to quantify the spectra under these DNP conditions, even in complex mixtures. Okay, so let's come back to that drugs in cells questions. Can we establish this concentration response relationship by seeing the drugs directly in cells? To do this, we use a protocol which is, let's say, emerging, becoming standard. This is what we came up with AstraZeneca. It's very similar to what Kendra Frederick and her group have come up with, and she has a very nice article that's already on the archive. So I strongly recommend that if you want to do this kind of thing, then follow the detailed instructions that they give you. Basically, in this case, and she's looking at, her group is particularly interested in looking at proteins for in-cell NMR. So maybe the difference that we do is that here we're just going to culture the cells in a medium containing 5 to 10 micromolar of the drug to incubate the cell in presence of the drug. Then we wash the cell to stop the drug uptake. We freeze in a buffer, in a protectant and store it at -80. We transport it from wherever we were to wherever we are. And then for the NMR experiments, you take them out of the fridge, you melt them at room temperature, 37 degrees. We spin them at 2000 G, discard the supernatant. We resuspend them in the DMSO D2OH2 mixture that we'd be using for DNP. Again, spin them, discard the supernatant, resuspend them now with a known amount of the DMSO D2OH2O solution, including the standard. Here we're going to be using histidine, and including the radical. Here we're going to be using Amipol. And then we pack the rotor by centrifugation, classically, close it with a silicon plug and cap, and then we directly freeze the rotor in the spectrometer. That's actually the main difference between what we do and what Kendra Frederick does. She uses a slow freezing protocol. The slow freezing protocol means that the cells might be viable after the experiment. The fast freezing tends to kill the cells, but what we see is that it doesn't destroy them, and that's what was important for us. And when we do NMR of the supernatant after the experiments, then we see no traces of lysate, so the cells remain intact. Okay, and here's a result. I mean, this was Andrea Bertarello's DNP moment. So this is the 15N spectrum of those cells. And we very clearly see the signal coming from the drug in this beautiful nitro group reporter, which is out there at 380 or so ppm, which is not overlapping with the rest of the cells. And so we very clearly can detect, using this reporter group, we can detect the drug very nicely using DNP MES with actually quite good signal to noise. So this is about 24 hours acquisition, and the cells were incubated with 10 micromolar drug for 6 hours before being washed and then frozen. And so now, of course, we'd like to determine the concentrations. So here's the case where we're incubating the cells with 5 micromolar drug for 6 hours. And again, there's a simple relationship between the concentration of the drug and the intensity of the NMR signals with a few potential unknowns. So we need to know, in this case, the enhancement of the histidine and the enhancement of the drug, if we want to make this quantitative. We also need to know the volume of the cells. The volume of the cells we can estimate. We have 3 million cells, so that should be 3 microliters. What we do know is the enhancement on the cells, and this from the proton spectrum, we can measure that. Can't measure the enhancements on the nitrogen because it's just too weak. So the cells show an enhancement of 25, and the DMSO, the solvent, shows an enhancement of 244. And remember, that's what we expect because the DMSO is the polarizing solution and the cells are being polarized by relay. Okay, so if we assume in the first model that the drug is distributed throughout the cell, then we can assume that the histidine enhancement is the same as the DMSO, it's outside, and that the drug enhancement is the same as the cells enhancement. Okay, and if we use that in that equation, we find a concentration of the drug of 171 micromolar. If we assume that the drug is located at the surface of the cells, so the drug for some reason is collected on the surface, then the enhancement of histidine would be the same as DMSO, and the enhancement of the drug would be the same as DMSO. And then if we plug that into this equation, we get a concentration of the drug of 18 micromolar. Okay, so we're going to have a range of concentrations that we can determine this way, which depends on what the distribution is. And hopefully we'll be able to find the distribution as a second step. And so we measure concentrations between 170 and 18 micromolar, in this case, of the drug in the cell. And here's a comparison of those 10 micromolar and 5 micromolar cases. And you can see that maybe as expected, when we increase the concentration of the drug in the incubation process, then we get an increased concentration in the final result here with, you know, roughly a factor 2 difference between the 5 micromolar and the 10 micromolar cases. And what's interesting here is that we're using the, in one case, we're using the histidine as an internal reference, but the internal reference, of course, allows us to calibrate the protein signals that we see from the cells. And so in the second case here, we're actually using the amides in the spectrum as an internal reference, okay? And of course, that also means that we can calculate the concentration of protein or amide in the cells as well as one of the side effects of these experiments. And here, for example, we find that the NH concentration is about 3 molar in these cells, and that agrees with what we expect from actually a very hazy literature, and what we can measure from mass spectrometry and things like that as well. So what's nice is that these numbers do seem to cross-validate with numbers that we can measure when we can measure them from elsewhere. So as I said, this is new for us. This is new work. We're quite excited about it. What I think I've been able to show you is that using a nitro group as a reporter, histidine as an internal standard, we can selectively both detect and quantify the accumulation of a drug in hex cells. As we notice, this is, let me just go back and remind you, this is an accumulation. We see a much higher concentration of drugs in the cells than we do in the incubating medium, and so there's active transport of the drug into the cell, and that was expected. The limit of detection for a nitrogen-15 DMP-MAS experiments is estimated to be about 2.5 micromolar, so about 50 picomoles in the sample. And the MAS DMP enhancements and build-up times hopefully we'll be able to yield the location of well, we know they can yield the location of substrates. And our next step obviously is going to try this to see if we can determine where the drug is located within these cells in the same way that we could do it for the nanoparticles that I showed you at the beginning of the talk. And so that's really all I had to say. Thanks to the people involved in this. Most of the work at APFL was done by Andrea Bertarello, who's a postdoc in the group, together with Pierrick Berrier, and they've been working very hard on this to get these results. And we've been doing this in collaboration with a group at AstraZeneca, so Stefan Schanz, who is in material science, and Elisabetta Chiapparina, two of the key players there. Elisabetta, again, is somebody who was a PhD student in Jeffrey's group back in the day, and their group, so in particular Marcus Schad and Urban Scans, who've been working directly in the project, and Chad Elmore and Sepida, who've been providing us with the drug and the cell samples. Thanks for your attention. Weather's getting better, and be happy to take any questions.
Thank you, Lyndon. I've been warned by Marcus that we have exactly 1 minute for questions. And in fact, as far as I can see in the Q&A, there is no other questions. So let me ask one or two. In the very beginning, you explained that you can make a factor of about 1 million in sensitivity. And then you also say that you have to do a decent reference experiment. But when you make this factor 1 million, you, among other things, suggest acquiring multiple FIDs by refocusing with some sort of Kappa cell sequence. Now, that's something that you could also do in the reference experiment, could you not? Is it, that's not specific to?
Not, yes, well, yes, no, that's true. In the million, the benchmark would have been a carbon-13 CP mass experiment. So if you, in that particular benchmark, so I didn't spend too much time talking about that. And so in the carbon-13 experiments, you wouldn't usually gain anything with CPMG. If you go to something like phosphorus or silicon or any of these kind of single nuclei, which are very easy to decouple, but which tend to have some heterogeneous broadening, then you'd gain a lot by CPMG. That's a good point, yeah.
And a similar question, you mentioned that you gain a factor 3 or so by dropping the temperature from 300 Kelvin to 100 Kelvin. Ludwig Boltzmann would agree with you, but you don't mention that there is also some line broadening when you freeze the sample. As far as I could see, that was not.
No, yeah, so in most of our samples, in most of the samples that we're interested in, there isn't any line broadening. So most of our samples are heterogeneous anyway, and so you don't have that extra line broadening. Yeah, but again, yeah, if otherwise you would need to include that line width term in those equations. And as you know, in the papers that describe, how to assess overall enhancements, there are a number of factors that weren't in my specific list that can also go in and go out. So T1, if the T1 is very long, then relay is very favorable because you're diffusion limited and not T1 limited, and things like that. So there's a whole bunch. I think my main message was to say there's a whole bunch of parameters that can be optimized to get to these very sensitive experiments as compared to what you would do if you just walk in on a Monday morning and try and get a CP mass spectrum. It's not just the DN, it's not just the epsilon.
Slightly different. This will be my last question. You mentioned that you can quantitate the concentration of protein by looking at the amide protons, if I remember correctly. But surely a protein is often a globular or folded protein, and then the intensity of the amide protons near the surface, which could easily be enhanced by DNP, would not be the same as from the bulk, from the interior of the folded protein. Do you take that into account, that the enhancement is not uniform?
No, we didn't. Again, of course, that's a very good question, but it's a subject that's a bit more debatable. In principle, the enhancement should be the same throughout the protein because you're getting your polarization from the frozen proton matrix, and the spin diffusion length scales are much bigger than the atomic length scales. So in principle, within a molecule, even if it's a protein, the enhancement should be uniform. And most of the experiments show, most materials show that property, but it's not always the case. There are some exceptions, and I don't think we understand very well when those exceptions occur. If those exceptions were occurring for those proteins in cells, for example, on those, then yes, that would have to be taken with it. So again, there are error bars on that measurement, of course. You noticed that.
Yeah. No,
in particular here, I put a kind of squiggly equals. I didn't even put 3 millimolar plus or minus anything because we're actually still working on really trying to validate or to confirm how accurate we think this should be, and also, of course, to make sure what that peak contains.
All right, thank you very much for your answers. I think the floor has to go back to Marcus. Thank you.
Thank you, Lyndon, for a fantastic presentation, and also Geoffrey for reminding all attendees that we have an unofficial part. So all the many questions which were put in the Q&A will be answered as part of the unofficial session. Yeah? So please hang on and we answer your questions, or Lyndon does so. So without further ado, then I want to move over to the second presentation. It's my great pleasure to introduce Nick Fawzi from Brown University. Nick undergraduated at University of Pennsylvania. He actually obtained 2 degrees, one in bioengineering and one in marketing. And then he moved on to Teresa Head Gordon's lab at UCSF and performed molecular dynamics simulations. And I guess that's where he fell in love with amyloid beta, because then the next, I don't know, at least 10 years, probably he worked on amyloid beta, first in Teresa Head Gordon's lab doing computations mostly, but then joining Marius Clore lab at NIDDK, and really doing fantastic work there in the development of, first of all, more probably application of paramagnetic relaxation enhancement, but then also developing dark exchange saturation transfer methods for probing dark states in biomolecular systems. From that on, he moved on then to an independent position at Brown University, where in 2019 then became associated professor. He also received the National Science Foundation Career Award, and is really, I mean, spearheading, I think, the NMR application to liquid-liquid phase separation. Yeah, putting us hopefully in a position to better understand the molecular details of phase separating intrinsic disorder proteins. So with that having been said, Nick, it's yours.
Thank you for the introduction and the opportunity to talk today. Like Marcus was saying, my group has focused on looking at phase separating proteins using solution NMR. And I want to start with the most important slide first, not the weather, but in this case, thank you to the group members who carried out this work, as well as the funders, and I'll try to highlight them, the group members along the way. So in my lab, we use solution NMR of disordered proteins to look at their behavior at atomic scale, residue by residue, and also combining that with some biophysical experiments, including microscopy, to see how do these disordered proteins self-assemble into these phase-separated interactions. And I guess I've graduated from looking at amyloid beta only to looking at this family of no more friendly proteins in terms of their behavior in vitro, but a large family of proteins called heterogeneous nuclear ribonuclear proteins, or HNRMPs. And these are RNA binding proteins that have significant regions of disorder in addition to their folded RNA binding domains. Many of these proteins have so-called low complexity or prion-like sequences that look like yeast prions. And these sequences are aggregation prone and devoid of most charge residues, as well as enriched in aromatic residues and polar residues. And an interesting thing for the NMR spectroscopist is that inside cells, these proteins are often ubiquitous in most cell types, but also in relatively high cellular concentration, some even up to micromolar concentration. So they make a good model. So, why might they be in such high concentration? Well, one reason might be is that they undergo this process of liquid-liquid phase separation, associating with themselves and with RNA and other proteins to form these membraneless assemblies. So this work from Paul Taylor that you see in the video shows one of these HNRMPs forming liquid assemblies in the cytoplasm, and then when stress is removed, these stress granules disappear or redissolve. And so these are dynamic things that flow and fuse and retain the spherical shape and can be assembled or disassembled. So the protein I'm going to talk about today, one of my favorites, is this protein fused in sarcoma. It's a RNA binding protein with this N-terminal low complexity domain, which we'll start out talking about, that is aggregation prone, but it also has 3 so-called RGG regions, or boxes, that are enriched in these arginine, glycine, glycine sequences that are also disordered or predicted to be disordered. And this protein is of interest because of its normal functions in RNA processing, but also it's mutated in ALS, and here's only a few of the mutations that are found in this aggregation-prone domain, as well as it's fused in a number of a whole family of cancers. And so here's just one example where the low complexity domain is attached by a chromosomal translocation to a DNA binding domain, and this results in this case, a sarcoma, but there's a number of related cancers that are caused by FUS and its paralogs. So what does this domain look like? Well, I've just taken the sequence of this first 163 amino acids and aligned it manually by showing you that there are these tyrosine repeats. So here's the first amino acid, here's the residue 163, and you see it has this unusual features, rich in serine, tyrosine, glycine, and glutamine, but lacking positively charged residues entirely, and have almost no aliphatic or negatively charged residues. So what does that mean? A number of years ago, we showed already that this protein is in fact disordered in solution. So it has this beautiful disordered spectrum. But if you cool down our NMR sample, you'd get it to be very cloudy. And we were wondering what that was. It seems like at the dawn of time in this work in phase separation, but when you look under a microscope, you see that if you increase the concentration or decrease the temperature for this low complexity domain of FUS, you can find it that it undergoes liquid phase separation. So at high concentration, you form these assemblies that are protein dense droplets. And so what we had done was we wanted to see, what does it look like? Can we use NMR to examine these liquid phases? And so if we made a very concentrated solution of the protein that is entirely unstable, it would form droplets. And we could actually take these droplets and collect them at the bottom of a large sample tube, in this case, like a 15 mil or 50 milliliter tube. And then we'd get a very small concentration at the bottom, or a small amount of protein at the bottom. And when we look at this state directly by solution NMR, we see that actually it looks very similar to the dispersed state in which the protein is essentially monomeric in solution. And so the fingerprint here by the proton nitrogen HSQC doesn't change that much. It is very highly concentrated in this, and our recent measurements actually suggest it's up to 20 millimolar, depending on the salt concentration and the temperature in this state. And you can, if you just record experiments kind of one-to-one, you can barely see the dispersed state. You have to zoom in by 100 times to see this signal for the dispersed state. So we're looking at the major species, and it's very concentrated. And the motions in this state are reduced. So we see changes in the transverse relaxation enhancements, as well as changes in the longitudinal relaxation. But we see that the protein is still relatively mobile because there's significant nanosecond reorientational motions, because the heteronuclear NOE for proton nitrogen is not dramatically changed. So what we've done more recently, I guess I'll skip ahead, is try to examine the state in more detail. And one current question is, how well do these giant macro droplets that we make actually represent these microscopic droplets that scatter light and are formed and are spontaneously formed in vitro? And so one way we sought to answer this question was to compare the spectra of our macroscopic condensed phase that we show here in green with samples where we actually have both phases present simultaneously. So a cloudy sample in which we have spontaneously forming droplets. And if we make that kind of sample, which is in red, we see that the spectra actually look like a superposition of the spectra of the dispersed phase in blue and the condensed phase alone in a sample like this in green. So for most of the spectrum, the resonances are overlapped. And so there's only a few places where we see significant enough difference, including at the very C terminus, which perhaps isn't surprising. It's experiencing a different chemical environment inside, or a significantly different chemical environment in the phase separated assembly. So we really don't see dramatic uniform shifts. I know this was a discussion we had with when Frederica Alain was presenting. So we don't see dramatic uniform shifts. We see some small uniform shifts and also some non-uniform shifts that suggest that there's a different chemical environment slightly inside this phase separated assembly. But we don't see dramatic changes in the folding or the structure. So, however, these proteins are highly crowded. This disordered domain is crowded in this state. And if we look at the diffusion NMR, we try to see how can we measure these diffusion coefficients. And so if we use kind of standard parameters in the dispersed phase, we see in blue that the FUS has a diffusion coefficient that gets dramatically slower if we measure in the condensed phase. So this is again in a single phase samples here. But if we really ask, well, are other things slowed down, we can see that the buffer even slows down dramatically from the dispersed phase to the condensed phase. So even small things are slowed when it's inside this phase. And if we really push the experiment to the limit, we can see that FUS diffuses slowly, again, in these PFG diffusion experiments. And so this diffusion coefficient for FUS is dramatically different in the phases. So then we tried to ask, well, what contacts are formed in these FUS low complexity domain phases? And this stuff is published, so I'll try not to spend too much time on this work, but we tried to use a number of different filtered edited experiments, as well as these doubly edited experiments to see which parts of the proteins are in contact in these phases. And I just want to point out, these are weak contacts. We don't think they're persistent, and they are formed between these proteins. Probably, I mean, I do not think that these contacts are going to be formed for anywhere near the length of time that the NOEs are going to build up. So these are, you can think about them as transient NOEs, but when we perform these experiments, we see that we can actually see noses from essentially all of the different positions in the FUS low complexity domain, all the different residue types to essentially all the different positions in the protein. And so here we're looking at NOEs measured starting from 15N labeled protein and ending on 13C labeled protein. And we can see that for protons attached to the C alpha of serine, we can see that we see NOEs corresponding to essentially all of the different regions in our nitrogen correlation spectrum. So it's interacting with glycine backbone, with asparagine, glutamine side chain, and with the other residue types as well. So we tried to rationalize, what does this look like by quantifying these NOEs, but essentially we see a broad distribution of different protons that are in contact. And, how can we see these protons? I was just calculating. So unlike, what Lyndon was saying here, we're looking at more like 10 micromoles of protein. And, for some of these amino acids like tyrosine, we actually have 24 of them. So, we're looking at, hundreds of micromoles of protein, or essentially 500 millimolar tyrosine in this sample. So these are very highly concentrated states. So that gave us some information about what amino acid types are in contact. And actually, there's been some really exciting work from Lewis K. and Julie Foreman K. using deuterated, partially deuterated samples or mixtures so that they can see which positions with higher resolution actually mediate contacts. But we wanted to take another approach using paramagnetic relaxation enhancement within the condensed phase to see which regions are in contact. And so when we perform these experiments with labels at 3 different positions, looking for intermolecular contacts, so we have a very small amount of this unlabeled or isotopically unlabeled and spin labeled protein, we see effects essentially across the entire domain. Unfortunately, the resolution here isn't what we would want it to be, so we're working on fixing that. But we see that we can see significant PREs that stretch across the proteins regardless of where we put these labels. So it suggests that contacts are not formed just between one part of this domain and another part of the domain, but they're formed across the domain. And that makes sense because the domain is highly repetitive in its sequence. So in the previous experiments, we identified a number of amino acids that were important, and in this field of phase separation, these aromatic residues and arginine residues and in other parts of FUS and in many proteins have been highlighted as being important in phase separation. But we saw a lot of NOEs to glutamine, so we asked, well, how does glutamine impact phase separation? And if we substituted glutamine with serine, we see that we can actually alter the amount of, or the extent of phase separation that we see. So in this experiment, we're making solutions of droplets, and we're spinning out those droplets with centrifugation. We're asking, well, how much protein is left in the supernatant? So the less protein left in the supernatant, the more phase separation is promoted by these different variants. And so we see here that adding more glutamines enhances phase separation. It also increases the partitioning of the protein into these phases. So we can see it by looking at the dispersed phase by itself, or we can see it by looking at the partitioning coefficient. And again, we think these contacts, in collaboration with Jatane Middle's group at Texas A&M, are weak. And so we've simulated these contacts, and we can see many different types of contacts between glutamine and other amino acids, mediated by not just the side chain, but also by the backbone. And so the picture that we have is that we can directly characterize this liquid state using solution NMR. and we can see that glutamine and hydrogen bonds contribute to phase separation. And in some work I didn't show using Hofmeister salt series, we show some evidence that hydrophobic interactions are also important contributors to the contacts that stabilize these phases. So this was work that focused essentially entirely on this low complexity domain, but the FUS is much larger and longer protein. As you can see, the low complexity domain here, this N-terminal domain, is only one part of the protein. So how do these other domains contribute to FUS phase separation, or liquid-liquid phase separation? And we know this is the case even from our earliest work on this area, that you can see robust phase separation of the low complexity domain at 200 micromolar, but at the same or similar conditions, the full-length protein phase separates robustly, actually at 1 micromolar or below, which is even the physiological concentration of FUS. So we wanted to probe, how do these other domains contribute to FUS phase separation? So we used a number of constructs to answer this, not only the full-length protein, but also the low complexity domain on its own, or one of these arginine, glycine motifs as a model for these RGG regions, as well as this version that lacked the low complexity domain in its entirety. And so what we can see is that if you mix the low complexity domain together with a version of FUS that lacks the low complexity domain, you can get phase separation at lower concentrations. Actually, even if you just add 5 micromolar of the of this form that lacks the low complexity domain, you can enhance FUS phase separation, which is not seen on its own until you get to 200 micromolar. So we think that there's bridging interactions between the low complexity domain and these other domains, especially these arginine glycine domains. And again, the blue data here refer to spots or positions in the solutions where we see droplets form. And if we mix the RGG domain with the low complexity domain, we again see enhancement of phase separation. So we asked, well, can we look at these assemblies using NMR spectroscopy? And so we made samples containing the low complexity domain and the RGG domain. Again, used a similar procedure to make large amounts. Again, this dilute phase is actually 50, maybe up to 50 milliliters of size, but we only load into the tube part of it above the condensed phase. And then the coil volume is, the concentrated phase is the only thing in the NMR coil. And if we look at that, we see that we get a nice spectrum for this 15N labeled RGG domain. We see that actually looks very similar to that in the dispersed phase, so that again, the disordered structure of this RGG domain is preserved in the dispersed phase, and really we don't see dramatic chemical shift perturbation. This RGG domain that we've selected has arginine glycine motifs here in this kind of central region and C-terminal part, as well as a glycine-rich N-terminal part. And we do similar kind of experiments here where we're looking at NOEs between 15N labeled RGG3 and 13C labeled low complexity domain. And if we use that labeling scheme, we can see that we see NOEs, again, essentially starting from essentially all the different amino acid types in the low complexity domain, or in that N-terminal domain here. They end up at essentially all of the backbone positions in the RGG domain. So here's a place where using deuteration, as Lewis and Julie have recently shown, would help these spectra dramatically to see which particular glycines perhaps, or which particular amino acids are enhanced more than others, but we see a relatively uniform enhancement. So interactions seem to be distributed throughout the entire sequence. We also wanted to see, well, how does FUS interact with its partners? And there's one partner that's been of interest to many people, this RNA polymerase II C-terminal domain. So the thing that makes mRNA, and we wanted to, it had already been reported that it interacts with FUS and the low complexity domain, and we show here that even the other domains of FUS, so FUS without the low complexity domain, can interact with that with that RNA polymerase II. If we make samples that contain all three of these disordered domains, we find that RNA polymerase II remains disordered inside this mimic of an environment where we have the different types of domains of FUS. And that again, we see NOEs from the low complexity domain or the RGG that highlight essentially all of the different residue types, including these unusual lysines in the RNA polymerase II C-terminal domain. And using some phase separation assays, we showed that disruption of these lysines actually alters the partitioning of the RNA polymerase II C-terminal domain into the phases of the FUS low complexity domain. So we think that these lysines have some contribution to interactions, some favorable contribution to interactions with the low complexity domain. So with that, I'd like to thank you for your attention, and thanks for the opportunity to speak here.
Thank you, Nick, for a very interesting presentation. So there is already a question, I guess. No, this is still from the previous one. So I can start while people type in the questions. So for the FUS droplets, I mean, do you see any evidence, let's call it, of aging, or how? I mean, because of course for the FUS there's this with amyloid formation. So how long can you measure before you see changes? Or
so that's a very good question. So when we made these macroscopic samples, they actually stay, you know, you might say good for weeks, month, even at least a month. So we don't see aggregation happening rapidly in this condensed phase. There may be some aggregation that forms at these interfaces, but for the FUS low complexity domain, we see a liquid form stay stable for weeks, which is quite surprising, I think. And it doesn't mean that this protein can't aggregate. We have other data that if you simply just agitate this low complexity domain, you get formation of aggregates really rapidly. So it suggests that the interfaces or the air-water interface, for example, induces aggregation. So this is an unusually stable state.
So that would, I mean, is it then on pathway, the phase separation to amyloid formation, or rather not?
I mean, I guess this would suggest that it's not on pathway. I mean, this is not going to, I don't know what happens in cells. And if we do use the full length protein of FUS, we see that it converts into aggregates rapidly, except if we phosphorylate the N-terminal domain. So we showed that previously that you can prevent aggregation by phosphorylation of this domain.
Okay, and then maybe I put a second one. Yeah, I mean, you did a lot of this exchange experiments. So did you try those on the condensates? And was there exchange or not?
Yeah, we tried extensively actually to see, are there minor species, minor states that we couldn't see by other techniques. So yeah, we tried, you know, are there fibrils? So we didn't see anything within the states by, you know, dark state exchange saturation transfer. So we could fit everything with a single directly observed state. We tried relaxation dispersion, but we didn't see any evidence of dispersion. Although I'm curious to use this off resonance R1 row that I think Lewis recently showed that in droplets of or condensed phase of DDX4, you see some intermediate timescale motion, but nothing with CPMG that we saw. And even spectral density mapping doesn't suggest that there's anything that we're missing. So we don't think that there is a significant minor structured state.
Okay, thank you. So then maybe I read the questions. So from Marcus Hennan, very cool talk. In the PRE experiment, what ratio do you use to mix the MTSL natural abundance sample with the 15N sample?
So for the PREs, we used about 1% labeled protein. When we made the sample, I can't guarantee that the partitioning coefficient isn't slightly different once you do this labeling. We were concerned that we didn't want a very high density of the spin label because the protein is essentially highly concentrated in this phase. So we use 1%.
Thank you. Philip Neudecker, just a technical question. What mixing times do you use for your nose experiments and how do you choose them?
So I don't have those data here on slides, but we did build-up curves, at least in 1 dimension and 2 dimensions, because these are filtered edited experiments or doubly edited experiments, and we could tell that they we don't see any change. So if we do very short mixing time, we don't see any difference in the kind of overall pattern. But we used I guess a variety of mixing times, but I have to say they were relatively long because they were these are not stable contacts. So
Zhongchun Munk, I hope. So I noticed that the LC region is so now it jumped. I'm sorry. So I noticed that the LC region is almost uncharged and the full length contains charges, arginine. Did you observe any differences in salt sensitivity, temperature sensitivity between the LC only, I guess, and the longer constructs of FUS?
This is a great question. So we're working on this actively now. There is a difference in the salt sensitivity. So for these low complexity domains that are devoid of charge residues, if you add salt, you get more phase separation, or you reduce the saturation concentration. So you add salt to get more phase separation, while the charged disordered domains that others have worked on, as well as these full-length proteins, if you add more salt, you get less phase separation because you're screening out interactions. And so what we have seen is that if you add the RGG domain, you get less phase separation with increasing salt. And then I saw the next question, is there common that phase separated material remains disordered? So everything that we've looked at, disordered domain remains disordered upon phase separation. So FUS low complexity domain, FUS RGG, C-term domain of RNA polymerase 2, HNRP-A2's low complexity domain, but with the exception of this helical region in TDP43 that becomes more helical due to helix-helix contacts upon assembly.
You maybe also want to answer the last from John Tainer, who's wondering if you would expect furamide, which resembles the glutamine side chain, to have the same impact as glutamine. So if you can mimic backbone H bonding capability in your experiment.
Yeah, I guess it would be interesting to see if you could essentially like screen out the interactions, because you would be removing, I guess, multivalent interactions by competing them with a essentially monovalent hydrogen bond former. It's akin to using salt to screen out interactions with a charged polymer. So that's a good question. We haven't tried that.
And then there's a last question regarding diffusion. I read this. Do you observe the difference between the expected hydrodynamic radius in solution versus the condensed phase? Probably one can extend this if there was sort of multi-exponential decay in the diffusion data or so. I mean,
so we don't see any multi-exponential decay, so everything can be fit with a single diffusion coefficient in the dispersed phase or in the condensed phase. We didn't try in the mixed samples because they're also, things are changing over time. Frederick Alain showed some nice data on, how do you stabilize those droplets using a gel. But in the dispersed phase or the condensed phase, we only have one species, it seems. And the question was, what was the question again? Do we?
I mean, I think it was sort of probably, I'm not 100% sure if I understood it. That's why I a little bit reformulated the question. But the question was whether there was a difference in diffusion coefficient between.
Oh, yeah, it's about hydrodynamic radius.
Yeah,
yes.
yeah.
So in the dispersed phase, the hydrodynamic radius looks reasonable, what you'd expect for a disordered protein that is, prone to self-interact. So we have published those values and compared it to like a folded protein lysozyme control. But in the condensed phase, so I don't think that hydrodynamic radius has a real meaning there, because essentially it's a polymer melt of these polymers. And so there's some debate. I think that these proteins will probably actually become more extended because they're somewhat collapsed in water. And so once they get into a happy place where they're surrounded by many copies of themselves, they'll be more extended. But that doesn't mean that they have a hydrodynamic radius. Their diffusion is going to be slowed because it's in a dense percolated system that's way beyond the overlap concentration. So these things are not going to have like a hydrodynamic radius.
Okay, thank you. With that, I would close the official part. Thank you again to both speakers for fantastic talks, and